So I really want to thank Joan and Julia for the kind invitation to come and speak today. I have to admit to feeling quite a bit daunted to speak today on the topic of anaerobic fungi, as I'm not an expert in this area at all, as you probably know, but I think of myself as more a rather enthusiastic supporter of research on this fascinating group of microbes. So I apologise in advance that my presentation will be a little light on the science today, particularly around the biology of the fungi themselves, but I did want to just touch on various activities that we have done in the area of anaerobic fungal research and connections that we've made that have been fostered by the network, which have been really excellent for us down here in New Zealand, where we are quite isolated from the rest of the world. And the network has been a great forum to connect us to global initiatives and meet new people and make exciting new collaborations, and to also just be able to generally help with the global effort to better understand the anaerobic fungi. So yeah, thanks Joan for your great work bringing us all together, and all those cool opportunities that have arisen from this. In my presentation today, I'm just going to start with a little bit of background on agriculture in New Zealand, and then a little bit of background about AgResearch. I'll then discuss a recent study where we were particularly interested in the impact of anthelmintic treatments on rumen fungi. And then I'll just give a little brief mention on a bit of work that we did a few years ago in cultivation and some of the connections that we've made since then within the network. So just moving on here. So firstly though, just as a little bit of background to farming in New Zealand, New Zealand has quite a temperate climate and quite fertile soils, and this supports almost year-round production on pasture-based farming systems for ruminants, which means that our diets here are relatively richer in fibre than some of the more intensive production systems used overseas, and hence we do have quite an interest in the fungi. Our economy here in New Zealand is highly dependent on agriculture. Primary products are responsible for almost half of our export revenue. And you can see here that we have nearly 40 million ruminants in New Zealand between cattle, sheep and deer alone. And that's in comparison to our human population, or our team of 5 million people. So with regard to AgResearch, some of you may or may not know us, but we are New Zealand's leading sort of Crown Research Institute, which is a sort of a government-owned research institute for agricultural science. And our sort of main mission is to deliver new knowledge and technologies that underpin New Zealand's pastoral, agri-food and agri-technology value chains. We have research centres and farms across New Zealand, with the largest sites based here at Grasslands in the North Island in Palmerston North, and the other large site is in Lincoln, which is near Christchurch in the South Island. The rumen microbiology research has been based traditionally at Grasslands, and this is where many of the folks who you may recognise from anaerobic fungal papers, such as Keith Joblin and Graham Naylor, and more recently Sandra Kittleman, who's been working with Peter Jensen. That's where they've been based and carried out a lot of research on anaerobic fungi in New Zealand. Both Keith and Graham have retired now, and as you might well know, Sandra's been based in Singapore for quite a number of years now. It's also been quite difficult to get research funding for anaerobic fungi here, as I've found. Methane mitigation is overwhelmingly the top priority for New Zealand, and most of the research investment is in rumen microbiology has been directed towards mitigating methane. But however, I've been sort of dabbling in this area whenever I can, and it does generally tend to slip below the radar a bit compared to other activities that we've been involved in though. But one study that I did want to present today was just from a recent project that I was involved in with Dave Lethwick, who's one of our principal parasitologists here at AgResearch, as well as Peter Jansen and Sandra Kittleman and several other authors as well. And this was basically to look at the effects of long-acting anthelmintics on the rumen microbiota and in the rumen fungi in particular. I think this audience probably doesn't need too much background information on the rumen, but just to briefly say that the rumen hosts a wide, quite a diverse and complex community of microbes, including bacteria, archaea, protozoa, and fungi, of course, also viruses. But these microbes collectively degrade plant-based feeds and they ferment them, providing energy substrates for the hosts. And the microbes themselves are also an important source of protein and lipid for the host, so they're important for nutrition in many ways. Methane is also a side product of this fermentation process. So just with regard to antiparasitic compounds, in New Zealand they're better known as drenches, or the anthelmintics. So in New Zealand, farmers commonly practice the blanket use of these drenches or anthelmintics during the pre-lambing season with ewes or female sheep. So they do this because they believe it improves the growth of both the ewes and the lambs will be heavier at weaning, which is more profit for the farmer. However, there's mounting evidence that these financial benefits are not always realised, and in addition, this excessive use of drenches is creating quite a major issue with regard to generating drench-resistant parasite populations. The drenches that are used, many of them have been reported to have antimicrobial activities. In particular, the benzimidazoles group of compounds contain some of the well-known fungicides. So one of our questions was, what are the impacts of these treatments on the rumen microbiota? So the aim of this study was to determine whether treatment of adult ewes with benzimidazole and or macrocyclic lactone long-acting treatments affected the composition of the rumen microbiota. And we had a couple of hypotheses aligned with these, and we thought that albendazole containing treatments, which are of the class benzimidazoles, will alter the rumen fungal community composition, and that moxidectin and abamectin, which are macrocyclic lactones, will alter the rumen bacterial community composition. Sorry, I've just flipped too many slides. So in the study, we conducted a sheep trial up at a farm in the Waikato with 300 ewes, and the animals were randomly assigned to treatments, either to one of two commercially available long-acting broad-spectrum drench products that are available in New Zealand, and then we had one group that was completely a control untreated group. Now, of the treatments, the first one, the CRC, stands for controlled release capsule, which dwells in the rumen and it slowly releases the abamectin into the rumen, which abamectin and albendazole into the rumen over 100 days. And then we have the LAI treatment here, which is a long-acting injectable product that contains moxidectin, and this is advertised to give control to parasites for over 100 days. And just with regard to that trial design there, we took rumen samples immediately before the animals were treated with those anthelmintics, and then we took rumen samples at days 35 and 77 as well. And sort of unlike what's shown in this diagram here, the animals weren't kept separately within their treatment groups. They were actually run as a single mob, so all treated the same. So with those rumen samples, we then used our standard primers to look at the bacterial, archaeal, and protozoal communities. And And then right around the time we were doing these analyses, the new Anaerobic Fungal ITS1 database was released, and that had lots of new taxa that were, I think, largely described from Mustafa El-Shahid's lab. And Sandra Kilman at that point kindly joined our group to design some new ITS1 primers based on these the new taxa that were available, and so we could better capture those new fungal taxa. And the details of the primers that we use are just shown here. So without going into too much details about other parts of the analyses, I'll just cut to the chase with regard to the fungi. What we did find with the slow release capsule treatment containing the ivermectin and albendazole through these ordination plots here, you'll see that over time, by day 77, the treatment had a slight impact on the fungal communities by that point, which was not what we expected from our hypothesis. But there did appear to be some separation there. But however, we did find that with that moxidectin injection, the fungal communities did separate reasonably clearly by day 77. And we don't know whether this separation is due to the compounds having a direct effect on the fungi themselves, or if they are modifying other parts of the microbial community that are then impacting the fungi. We're not too sure what is going on there, but just a summary of the changes in general that we saw through the different groups. The largest change that we actually saw was with the archaea, with that controlled release capsule. They showed the most drastic changes in community composition. The injection, the moxidectin, actually affected the bacterial communities early on, and then at day 77 as well. And then we saw that result on the fungi that I just showed you. So we're just interested to look at the abundance correlation relationships between the different microbial taxa, just to see how things sort of correlated with each other. And this slide here just really zooms up onto the fungal taxa that showed that showed to have some significant correlations. And you can see here the red lines indicate positive correlations, with the intensity reflecting how intense that correlation was, and the blue lines reflecting negative correlations. And you can see that the treatments, the drench treatments did alter those relationships somewhat. And for example, you can see here with the injection treatment that the fungal taxa started to show some positive and negative correlations with members of the Ruminococaceae. And it's sort of tempting to speculate that they may be competing for similar niches in terms of fibre degradation in the community, and potentially, moxidectin might be having an effect, potentially affecting the dynamics between the microbes here, but that's purely speculation. There clearly needs to be a lot more investigation to see if there is actually anything really happening there. But just sort of to conclude this bit of the work, we found that those long-acting antiparasitic compounds did have small but detectable impacts on the rumen microbial communities, and the extent of those impacts really varied by the microbial group, where, as I showed, the archaea seemed to be the most impacted, followed by the bacteria, then the fungi. Protozoa, we didn't actually really see any changes in, and they're usually quite a variable group anyway. So what is interesting is whether there is a biological impact on the animal or on the community due to these changes in microbiota structure, and that is something that we haven't had the opportunity to look into yet. So I just wanted to switch tack now to mention actually quite an enjoyable time we had a few years ago in our lab. So at the time, our most of the rumen micro team was really heavily into omics type investigations, and bacterial and archaeal cultivation was ongoing, largely as part of the Hungate 1000 study, but the cultivation of rumen fungi had become somewhat of a lost art, and especially since Graham Naylor had retired, I think it was about 2010 or so. So we were really interested in relearning some of the old techniques and then getting back into the lab. And I managed to find a little bit of funding to kick this off. So we managed to convince Graham to come in from his retirement and come into the lab and teach us. So Graham came in and he was teaching myself Priya Soni, who you see pictured here, and Rene Atua, isolation and cultivation methods using the old agar roll tubes and sloppy agars, sort of methods he'd worked up with Keith Joblin back in the day. And we also managed to dig up his favourite special old microscope that he used to use for looking at fungi, which was luckily still hiding in the corner of the lab and hadn't been dumped out. So we set to sort of digging into some of the old cultures in the freezer that he worked with Keith, and we also performed some isolations from sheep and cattle on fibrous diets as well, just to get our hand in there and have a go. And we did manage to get a number of isolates out from that little investigation there. Then we went on to try and identify what we had using molecular methods, and we naively went in using ITS primers, and soon realised that we ran into these well-recognised problems that you guys all know about with getting linked sequence heterogeneity from our standard sort of ABI Sanger reads, and for several isolates, and going, oh, how do we get around this? Do we have to clone all these copy numbers and things like that? But so Priya, she applied for a little bit of internal funding, and she managed to get some to follow up on this issue, where she was going to try and delve into separating these copies. And this was around the time that the HypoAF opportunity came through the network, which was a bit of a call for pure cultures to help populate the new anaerobic fungal ribosomal RNA sequence databases. So we got in touch with Deanna and Veronica to see if the program would be interested to work with our isolates, and it ended up that they really kindly agreed to do so, and we actually arranged for Priya to go over to their lab in Germany and work with them on it, so she could learn some of these methods and help work on the New Zealand isolates with them. And then unfortunately COVID hit, and the borders all shut. So unfortunately, Priya didn't manage to get over to Germany in the end and visit the folks at the LVL. So she ended up sending the DNA there to Diana, who's been working on the strains there. And yes, we're yet to catch up with Diana about the results in detail, but from what I hear so far, there's some nice nice strains here that we can find in our New Zealand animals. So another sort of global call that we caught wind of through the network was actually the Global Herbivore Microbiome project that's led by Mustafa El-Shahid at Oklahoma State University, and they were seeking samples from around the world to examine fungal communities. So we also got in touch with Mustafa and arranged to send them samples that we could collect from a number of host species in New Zealand. So we managed to collect samples from cattle, deer, sheep, goats and horses, and these turned out to be from colleagues at AgResearch who we knew had hobby farms or pet animals, because it turned out that you needed special animal ethics approvals even to collect samples from the ground, which was what we were planning to do, but we managed to get enough willing participants to collect from the animals at home. So we extracted DNA and sent those over to Mustafa. But in addition, our research team Grasslands had previously done a survey of the rumen microbial communities from around the world, and this was in a project that was led by Gemma Henderson and Peter Jensen that we call the Global Rumen Census. In this study, Gemma and Peter examined the rumen microbial communities of over 30 different animal host species and from over 35 countries around the world. So that was around something 700 odd samples from collaborators all around the world contributed to the study. And Gemma examined the bacterial, archaeal and protozoal communities, but unfortunately she didn't manage to report on the fungal communities. They actually had great technical difficulties getting the amplifications going with the ITS region that they were working with. So at the end of the day, they decided to leave this component out. And I was always really very sad about that because I thought it's an important part of the community. It'd be really interesting to know how that sort of correlates with the rest and interacts with the rest of it and the different diets as well. So when we did catch up with Mustafa, we did also explain about this collection that we had here, and he was rather excited about the collection and offered to take a look for us. So we've since got the DNA back out of the freezer or re-extracted where we've needed to, and contacted all the collaborators to get their permissions to use the DNA again so that we can hopefully find out more about this missing component of the community. So yeah, we really especially using the newer techniques and reference databases that are being generated these days. So yeah, we really look forward to gaining more insight into those relationships between the fungal taxa, the diet, the host, and the rest of the microbiome. And I'd also just like to mention here as well that with this global rumen census collection, currently we are in the process of sequencing the metagenomes of these with our collaborator, Dr. Xinguo Zhao at the Chinese Academy of Agricultural Sciences. And once this is done, these data will become available to the public as well. So they might be able to provide further insights into the rumen anaerobic fungi, depending on how much depth we get there with that sequencing. So yeah, just with that, I'd just like to end by acknowledging all the people on this slide here, which there are quite a few, but particularly Peter Jensen and Dave Lethwick, who I mentioned were involved in the anthelmintic study there, and Peter, of course, for the global rumen census work, and the many people that was also involved in the anthelmintic study. Really a huge thanks to Graham Naylor, who was generous with his time in coming in to help out Priya, Renee and myself there. Certainly a big thanks to Sandra Kuttleman for her help with guiding our analysis of fungi in the anthelmintic study. Deanna and Veronica for taking some of our strains and looking into the IDs for us. Mustafa, Noha Youssef and Radva Hanafi for the collaborations with the global herbivore microbiome, and Xinguo Zhao, who I just mentioned before. And we have our funders here as well. So yeah, thanks very much for your attention. Cheers.